So why is it so important to learn about protein structure? Well, let's take the example of Alzheimer's disease, which affects the brain. So in certain people as they age, proteins in their neurons start to become misfolded and then form aggregates outside of the neurons, and this is called amyloid. So amyloid is really just clumps of misfolded proteins that look a bit like this. And as you can see, as this amyloid builds up, it starts to interfere with the neuron's ability to send messages, and this leads to dementia and memory loss. So if we can understand how these proteins become misfolded in the first place, then we might be able to find a cure for this debilitating disease. And to understand how proteins become misfolded, we must first understand how they become properly folded. So before we begin, I just want to do a quick review of terms. You can have one amino acid, so I'll just write AA for amino acid, and then you can have two amino acids that are linked together by a peptide bond. So this is a peptide bond. And as you add more and more amino acids to this chain of amino acids, you start to get what is called a polypeptide, or many peptide bonds. And each amino acid within this polypeptide is then termed a residue. And then proteins consist of one or more polypeptides. And so I will use the terms polypeptide and protein interchangeably. So at the most basic level, you have primary structure. And primary structure just describes the linear sequence of amino acids. And it is determined by the peptide bond linking each amino acid. So if I were to take my amyloid example from Alzheimer's disease, and I stretch out that protein all the way, then this linear sequence is just the primary structure. So then moving on, we have secondary structure. And secondary structure just refers to the way that the linear sequence of amino acids folds upon itself. This is determined by backbone interactions. And this is determined primarily by hydrogen bonds. There are two motifs or patterns that you should be familiar with. The first of which is called an alpha helix. And if you were to take this polypeptide and wrap it around itself into a coiled-like structure, just like so, then you would have the alpha helix. And the hydrogen bonds just run up and down, stabilizing this coiled structure. And another motif or pattern that you can be familiar with is with a beta sheet. And that just looks like this. It kind of looks more like a zigzag pattern. And the beta sheet is stabilized by hydrogen bonds, just like so. And if you have the amino ends and the carboxyl ends line up, like so, then this sheet is called a parallel beta sheet. And then conversely, if you have a single polypeptide that is then wrapping up upon itself, just like this, and you have the hydrogen bond stabilizing like so, then you have the amino end coming around and lining up with the carboxyl end, and you have a anti-parallel configuration. There is a third level of protein structure called tertiary structure, and tertiary structure just refers to a higher order of folding within a polypeptide chain. And so you can kind of think of it as the many different folds within a polypeptide, which then fold upon each other again. And so this depends on distant group interactions, so distant interactions. And just like secondary structure, it is stabilized by hydrogen bonds. But you also have some other interactions that come into play, such as van der Waals interactions. You also have hydrophobic packing, and also disulfide bridge formation. So if we explore hydrophobic packing just a little bit more over here, say we have a folded up polypeptide or protein. And this protein is found within the watery polar environment of the interior of a cell. So if we have water on the exterior of this protein, then we will find all of the polar groups on the exterior interacting with this water. And then on the interior, you would find the nonpolar or hydrophobic groups hiding from the water. Disulfide bridges, on the other hand, describe an interaction that happens only between cysteines. So cysteines are a type of amino acid that have a special thiol group as part of its side chain. And this thiol group has a sulfur atom that can become oxidized. And when this oxidation occurs, you get the formation of a covalent bond between the sulfur groups. The formation of a disulfide bridge happens on the exterior of a cell. And you tend to see the formation of separated thiol groups on the interior of a cell. And that is because the interior of the cell has antioxidants, which generate a reducing environment. And since the exterior of a cell lacks these antioxidants, you get an oxidizing environment. So if I were to ask you which environment favors the formation of disulfide bridges, you would say the extracellular space does. Then there is one final level of protein structure, and that is called quaternary structure. And quaternary structure describes the bonding between multiple polypeptides. Multiple polypeptides. The same interactions that determine tertiary structure play a role in quaternary structure. And so let's say I have one folded up polypeptide, two folded up polypeptides, and a third and a fourth. The quaternary structure is described by the interactions between these four polypeptides. And within the completed protein structure, each individual polypeptide is termed a subunit. Since this protein has 4 subunits, it is called a tetramer. And so if I were to have 2 subunits, it would be called a dimer, 3 would be called a trimer, and then anything above 4 is called a multimer. So the term for a completely properly folded up protein is called the proper conformation of a protein. And to achieve the proper conformation, You must have the correct primary structure, secondary structure, tertiary structure, and quaternary structure. And if any of these levels of protein structure were to break down, then you start to have misfolding, which can then contribute to any of a number of disease states. Let's talk about conformational stability and how this relates to protein folding and denaturation. So first, let's review a couple of terms just to make sure we're all on the same page. And first, we'll start out with the term conformation. And the term conformation just refers to a protein's folded 3D structure, or in other words, the active form of a protein. And next we can review what the term denatured means when you're talking about proteins. And denatured proteins just refer to proteins that have become unfolded or inactive. So all conformational stability is really talking about are the various forces that help to keep a protein folded in the right way. And these various forces are the 4 different levels of protein structure. And we can review those briefly right here. So recall that the primary structure of a protein just refers to the actual sequence of amino acids in that protein. And this is determined by a protein's peptide bonds. And then next you have secondary structure, which just refers to the local substructures in a protein. And they are determined by backbone interactions held together by hydrogen bonds. Then you have tertiary structure, which just talks about the overall 3D structure of a single protein molecule. And this is described by distant interactions between groups within a single protein. And these interactions are stabilized by van der Waals interactions, hydrophobic packing, and disulfide bonding, in addition to the same hydrogen bonding that helps to determine secondary structure. And then quaternary structure just describes the different interactions between individual protein subunits. So you have the folded up proteins that then come together to assemble the completed overall protein. And the interaction of these different protein subunits are stabilized by the same kinds of bonds that help to determine tertiary structure. So all of these levels of protein structure help to stabilize the folded up active conformation of a protein. So why is it so important to know about the different levels of protein structure and how they contribute to conformational stability? Well, like I said, a protein is only functional when they are in their proper conformation, in their proper 3D form. And an improperly folded or degraded, denatured protein is inactive. So in addition to the 4 levels of protein structure that I just reviewed, there is also another force that helps to stabilize a protein's conformation, and that force is called the solvation shell. Now, the solvation shell is just a fancy way of describing the layer of solvent that is surrounding a protein. So say I have a protein who has all these exterior residues that are overall positively charged. And picture this protein in the watery environment of the interior of one of our cells. Then the solvation shell is going to be the layer of water right next to this protein molecule. And remember that water is a polar molecule, so you have the electronegative oxygen atom with a predominantly negative charge, leaving a positive charge over next to the hydrogen atoms. The same is true for each of these water molecules. So now, as you can see, the electronegative oxygen atoms are stabilizing all of the positively charged amino acid residues on the exterior of this protein. So as you can see, the conformational stability of a protein depends not only on all of these interactions that contribute to primary, secondary, tertiary, and quaternary structure, but also what sort of environment that protein is in. And all of these interactions are very crucial for keeping a protein folded properly so that it can do its job. Now, what happens when things go wrong? How does a protein become unfolded and thus inactive? Well, remember that this is called denaturation, and this can be done by changing a lot of different parameters within a protein's environment, including changing the temperature, the pH, adding chemical denaturants, or even adding enzymes. So let's start with what happens if you alter the temperature around a protein. And we can use the example of an egg when we put it into a pot of boiling water, because an egg, especially the white part, is full of protein. And this pot of boiling water is representing heat. And remember that heat is really just a form of energy. So when you heat an egg, the proteins gain energy and literally shake apart the bonds between the parts of the amino acid chains. And this causes the proteins to unfold. So increased temperature destroys the secondary, tertiary, and quaternary structure of a protein, but the primary structure is still preserved. So the takeaway point is that when you change the temperature of a protein by heating it up, you destroy all of the different levels of protein structure except for the primary structure. So now let's say you were to take an egg and then add vinegar, which is really just an acid. The acid in the vinegar will break all the ionic bonds that contribute to tertiary and quaternary structure. So the takeaway point when you change the pH surrounding a protein is that you have disruption of ionic bonds. And if we think about this a little bit more deeply, it kind of makes sense because ionic bonds are dependent upon the interaction of positive and negative charges. So when you add either an acid or a base, which in the case of an acid is just like adding a bunch of positive charges, you kind of disrupt the balance between all of these interactions between the positive and negative charges within the protein. So now let's look at how chemicals denature proteins. Chemical denaturants often disrupt the hydrogen bonding within a protein. And remember that hydrogen bonds contribute to secondary, tertiary, all the way up to quaternary structure. So all of these levels of protein structure will be disrupted if you add a chemical denaturant. So let's take our same example of a protein with an egg. And say if you were 21 years or older, you got your hands on some alcohol and you added this to the egg. Then all of the hydrogen bonds will be broken up, leaving you with just linear polypeptide chains. And then finally, let's take our hard-boiled egg from the temperature example, and let's eat it. So here's my beautiful drawing of a person representing you eating this hard-boiled egg. Once the egg enters our digestive tract, we have enzymes that break down the already denatured proteins in the egg even further. They take the linear polypeptide chain, whose primary structure is still intact, and they break the bonds between the individual amino acids, the peptide bonds, so that we can absorb these amino acids from our intestines into our bloodstream, and then we can use them as building blocks for our own protein synthesis. And that's how enzymes can alter a protein's primary structure, and thus the protein's overall conformational stability. So what did we learn? Well, we learned that the conformational stability refers to all the forces that keep a protein properly folded in its active form. And this includes all of the different levels of protein structure as well as the solvation shell. And we also learned that a protein can be denatured into its inactive form by changing a variety of factors in its environment, including changing the temperature, the pH, adding chemicals or enzymes.